Hello, today's video is on epigenetics and its application and benefits. In particular, this particular test called Epigene. Epigenetic testing. Epigenetic testing offers several benefits, including personalized insights to your own health because it's personalized, early disease risk assessment, and tailored lifestyle recommendations that will come with each report. It can review how lifestyle factors and the environment influence gene expression, leading to a better understanding of individual health risks and opportunities. This knowledge can empower individuals to make informed choices about their health and wellness. Epigenetics is the study of how your behaviors and environment can cause changes that affect the way your genes work. Unlike genetic changes, epigenetic changes are reversible and do not change your DNA sequence, which you inherit from your parents, but it can change how your body reads a DNA sequence. Gene expression refers to how often or when proteins are created from the instructions within your genes. Your genes are encoded to do many things to run the body, but it's whether how to make for them to work. They have to make proteins to go there and actually affect the tissues and organs. While genetic changes can alter which protein is made, epigenetic changes affect gene expression to turn on the genes or off. Okay, that's epigenetics. You're turning the genes on or off. Since your environment and behaviors, such as diet and exercise, can result in epigenetic changes, it is easy to see the connection between your genes and your behavior and environment. How does epigenetic works? Epigenetic changes affect gene expression in different ways. Types of epigenetic changes includes DNA methylation. DNA methylation works by adding a chemical group as a CH2OH, CH2, to the DNA. Typically, this group is added to specific places along the DNA where it blocks the protein to attach to DNA to read the gene. Okay, once it's methylated, you cannot read the gene. The chemical group can be removed through a process called demethylation. Typically, methylation turns off the gene and demethylation turns the gene on. How epigenetic works, another way is by histone modification. DNA, it's not just a straight shoelace, isn't it? It's folded and it's wrapped around proteins called histones. When histones are tightly packed together, proteins that read the genes cannot access the DNA as easily, so the gene is turned off. When histones are loosely packed, more DNA is exposed or not wrapped around a histone and can be accessed by proteins that read the genes. So the gene is turned on. Chemical groups can be added or removed from histones to make the histones more tightly or loosely packed, turning the genes off or on.
Another epigenetic process is histone modification. The long coils of DNA in our cells are wrapped around clusters of proteins called histones. How loosely or tightly they're wound around these histones controls how little or how much of the gene can be read and expressed.
So, while DNA methylation is...
There you go. See, if the histones, the proteins, are tightly wrapped, you cannot read it. The methylation of DNA and histones causes nucleosomes to packed tightly together. Transcription factors cannot bind to the DNA and genes are not expressed. But when it's loosely packed, the genes can be read and can be transcribed, and the genes can be expressed. Okay, why Epigene DNA methylation epigenetic tests? Firstly, it's accurate biological age measurements, 17 different age-related insights, prestigious collaborations with Yale, Harvard, Duke, and Columbia universities to develop this array of tests, high precision and reliability, powered by the latest science utilizing second and third generation epigenetic clocks, certified by CLIA and HIPAA compliant. It gives you predictive and preventive health insights. So it's personalized and actionable insights. They'll give suggestions in each report which part of your health you can improve, different nutrition, different lifestyle adjustments that you can make. Okay, so Epigene, there are 12 checkpoints, okay, that we measure your health status, your biological age, etc., and your health risk. Okay, I'll go to each individual checkpoint individually. Okay, checkpoint 1, we use something called BioAge. And why did we choose BioAge? Unlike traditional BioAge tests that focus solely on DNA methylation, BioAge leverages multi-omics to analyze aging from multiple perspectives. So what is multi-omics? Multi-omics refers to the integration of data from different biological domains to understand how they interact and influence health. So it includes genomics, transcriptomics, metabolomics, epigenomics, proteomics, and phenomics. Okay. By combining these layers, BioAge creates a comprehensive snapshot of your biological aging process, offering unparalleled accuracy and depth. This integration allows for personalized recommendations that target not just symptoms, but the root cause of aging. Checkpoint 2, organ age. The idea behind organ age is a unique tool that looks at how aging is affecting each major system in your body. Developed by top scientists in collaboration with Yale University, OrganAge uses advanced analysis to give you a detailed map of your body's aging journey. It's like having a personalized guide to understanding which parts of your body might need a little more attention to stay healthy and young. There you go. Patient is actually 43, but organ age appears to be 45. But individually, like the lung is younger, the brain is 46, so it gives you an age for each organ. Organ age uses information from your DNA to figure out how each of your organ system feels. Checkpoint 3, your Dunedin pace. Think of it this way, your biological age is like the distance you've traveled on a road trip. But Dunedin pace is your speedometer. It measures how fast are you aging, okay? Dunedin pace uses 173 specific DNA markers to measure your speed of aging. For instance, if you have a score of 1.0, it means your biological age, okay? Your biological aging matches your chronological age. You're aging 1 biological year for every year you lived. If it's more than 1, of course, it's bad. Your body is aging faster than average, which may indicate there are areas in life where lifestyle adjustments could help slow down your aging. If you're aging less than 0.1, that means it's good. Like 0.95 means you are aging 0.95 biological year for every year that you live. If you Google Rejuvenation Olympics, you'll find that Dunedin pace is the test they use to see who aged the slowest, okay? Brian Johnson, a renowned person in longevity, is aging at the age of 0.54. So he's aging the slowest. So now rich people are competing at not how much wealth they have, but how healthy they are and who has the slowest rate of aging. Checkpoint 4, immune health. Your immune system works tirelessly to protect you from infections, diseases, and even cancer. But just like the rest of the body, it ages as well. The process is called immunosenescence. Okay, your immune cells become senescence, and it can weaken your body's ability to fight off new threats. As you age, changes in DNA methylation, the process that controls which genes are on or off, can reduce the numbers and effectiveness of certain immune cells. So cells we test for is your naive CD4 T cells, memory CD4 cells, naive CD8, okay, these are T cells, basophils, B cells, memory cells, naive B cells, regulatory T cells, eosinophils, natural killer cells, neutrophils, and monocytes. Okay, checkpoint 5 is telomere, your telomere length, which is basically a gauge of your cellular aging. Telomeres are like protective caps at the ends of your DNA strand. I mean, your DNA, okay, it's a long strand. It's like the plastic tips at the end of the shoelace to make sure the shoelace stays intact, okay? Their job is to protect your chromosomes and to keep your genetic information stable. Without telomeres, your DNA could unravel or become damaged, which could lead to serious health problems. So in the report, you will get basically a measurement of how long your telomeres are, okay? And from then, they can predict a estimated biological age for yourself. Here you go, telomere at the end of your DNA, or end of your chromosomes. So telomeres are part of our DNA and has been shown that their length is directly related to aging. Every cell in the body has a nucleus which holds the chromosomes. Here you go, the nucleus of the cell. That's the chromosome, and there you go, at the end of the tips of the DNA. The chromosomes hold genetic information related to our cells. When a cell divides, the chromosomes are replicated to make sure the cells also hold all the necessary genetic material. So when a cell divides, okay, it makes exactly the same replica of information in the DNA in the new cell. The telomeres are made of about 15,000 bases at conception when you're born, right, of a new human. Each time the cell replicates, it shortens a bit. So when your cells replicate, divides, it shortens and shortens and shortens. The process of replication shortens the telomeres. If there were no telomeres, the DNA would be shortened and the vital information would be lost and life would not be possible. So telomeres play a very important role in providing protection to DNA. When they reach a length of 5,000 bases, the cells begin to die and eventually we all die as well, right? So when you're born, 15,000 base pairs. So it keeps dividing, your cells keeps dividing as you live. Each time the cells divide, the telomere shortens, and when they reach about 5,000 base pairs, the cells are not viable and they die. There you go, the telomeres. When you're born, 15,000 base pairs. As you age, you just get shorter, 10,000, 7,000, down to 4,000, 5,000, and then the cells die, and then we die.
What are telomeres? Telomeres are structures made from DNA sequences and proteins found at the ends of chromosomes. They cap and protect the end of a chromosome like the end of a shoelace. In almost all animals, from the simplest to the most complex, telomeres are required for cell division. With each cell replication, the telomeres get shorter and shorter until they're so short that your cells can no longer divide. When cells no longer divide, tissues age. However, telomeres can be rebuilt by an enzyme called telomerase to restore cell division. If you think we've found the fountain of youth, think again. If a cell keeps dividing uncontrollably and overcomes the limitations of telomeres, a cancerous tumor can form. This is why telomeres are so important. In healthy cells, they achieve the necessary balance between limiting cellular lifespan and your own longevity and keeping cancer growth at bay.
Okay, telomerase is the enzyme telomerase has the ability to lengthen the telomere by synthesizing DNA from an RNA primer. So some anti-aging researchers and scientists believe that by using nutrition or lifestyle changes, you can lengthen your telomeres and thereby lengthen your longevity. Okay, so it's an enzyme called telomerase. Nutrition and telomere. To understand how nutrition helps to improve telomere health and length, we must understand what is methylation. This is the process of donating a methyl group, a CH3 molecule, to the genetic material of the telomere. This process marks the telomeres for proper and health and functioning of the telomeres. In order to achieve this methylation, the body needs methyl donors. One methyl donor is S-adenosyl L-methionine, or SAMe. SAMe is available as a supplement, but it's better to let your body build it naturally using the building block nutrients such as methionine, MSM, sulfur, choline, and trimethylglycine. There you go. This is a SAMe supplement which you can buy in most pharmacies. You can also get all these nutrients from a good multivitamins along with some sulfur-rich protein such as whey protein, eggs, cottage cheese, dairy, red meat, chicken, legumes, duck, nuts, seeds, and eggs contain more choline than any other food source. Other nutrients, minerals and antioxidants are also important nutritional elements for healthy telomeres. Antioxidants reduce the amount of free radicals which can damage the telomeres. Magnesium is important for enzymes that help in DNA replication. Zinc is another mineral that helps DNA repair. Carnosine, I take zinc carnosine daily. It's an antioxidant that contains zinc and it's been shown to reduce the rate of telomere depletion in human fibroblasts and extend their longevity. It's also a good brain antioxidant. Vitamin C has been found to show the slow loss of telomere length in human endothelial cells. Vitamin D fights inflammation and supports telomere length. Vitamin E, especially tocotrienols, has been shown to actually restore telomere length in human fibroblast cells that also help in restoring damaged DNA. Other nutrients that preserve telomere length are quercetin, green tea catechins, grapeseed extracts, curcumin, resveratrol, grapeseed extract, and curcumin are especially useful because they help in generating longer telomeres. Factors affecting telomere length: eating junk food, smoking, sedentary life can lead to shorter telomeres. Stress and infections can lead to inflammations and hence shorter telomeres. The gene that causes inflammation is known as NF-kappa B. Nutrients such as those found in fruits, vegetables, and nuts and whole grains can actually calm this signal and reduce inflammation. Caloric restriction, like intermittent fasting, can lengthen telomeres. Eating excessively on a regular basis shortens telomeres. But caloric restrictions activate SIRT1 gene, which increases mitochondrial biogenesis, stimulates your mitochondria, thereby contributing to a healthy, increased lifespan, and reducing age-related disease. These can lead to longer telomeres, and resveratrol also activates the SIRT1 gene. High-intensity exercise is a good way of improving your telomere health and length. Okay, so now it leads to checkpoint 6, mitotic clock. Mitotic clock means how fast your cells multiply, cellular turnover. Every cell in our body has a job to do, whether it's repairing damaged tissues, replacing old cells, or keeping your organs running smoothly. To do this, cells divide, a process called mitosis. Okay, when your cells divide, it's called mitosis. The mitotic clock measures the number of time your stem cells have divided over time. So you get a report, okay? Effects of increased or uncontrolled mitosis. If it's beyond control, it will accelerate aging. Increased mitotic activity leads to faster cell turnover, but it also uses up the regenerative capacity of your stem cells more quickly. Over time, this can reduce your body's ability to repair itself, contributing to signs of aging like wrinkles, slower healing, or declining organ function. So if you have reduced regenerative capacity, maybe it's time to have a bit of a cell therapy, okay? Live cell therapy. DNA damage and mutations. Every time a cell divides, there's a chance for errors in DNA replication. When mitosis happens too frequently or is uncontrolled, these errors can build up, increasing the risk of mutations. And when you have more mutations, the risk of cancer or what they call it, whether it's benign or malignant growth can occur. Increased cancer risk. Uncontrolled mitosis, where cells divide without regulation, is a hallmark of cancer. When cell bypass the body's natural checks and balances, they can form tumors and spread into other parts of the body. And chronic conditions can lead to chronic conditions. Tissue with high mitotic activity may become more prone to conditions like inflammation or fibrosis, okay, which can affect the health of an organ over time. Okay, so the role of DNA methylation in stem cell division. DNA methylation plays a crucial role in controlling mitosis. It regulates which genes are turned on or off, ensuring that stem cells divide at the right time and the right rate. Okay, not too fast, not too slow. Okay, so DNA methylation regulates gene mitosis. Okay, so I won't go into it. Basically, methylation protects the integrity of your DNA during cell division by stabilizing chromosomes and preventing errors. When the regulation falters, it increases the risk of mutations, potentially leading to accelerated aging or disease like cancer. Okay, and controls your stems. DNA methylation maintains the identity of stem cells by controlling the expression of genes that determine the ability to differentiate into specialized cells. Okay, maintains ability to actually differentiate into specific cells needed. Disruptions in gene methylation can lead to stem cells losing their function or dividing too quickly. And regulating DNA methylation also regulates cell division. Proper DNA methylation ensures a balance between quiescence, when the stem cells are at rest, and division. So it regulates when the cells should rest, stem cells should divide, or just stay quiescent. If methylation patterns becomes abnormal, it can result in excessive cell division, depleting the stem cell pool over time. Checkpoint number 7, inflammation. As you age, epigenetic changes, including DNA methylation, can disrupt your body's how your body regulates inflammation. These changes can increase the activity of pro-inflammatory markers such as CRP, C-reactive protein. If it's high, it means you're having inflammation, and interleukin 6, which are linked to age-related disease and cognitive decline. Okay, here's a report. Measures your DNA methylation, CRP, and DNA methylation, interleukin 6. DNA methylation plays a critical role in regulating these markers, providing a more accurate picture of inflammation than traditional tests. You can take a blood and just measure CRP or your interleukin 6, but this methylation way of measuring them is more accurate picture related to disease. I'll show you. Okay, how DNA methylation inflammation tests are better than the conventional testing of CRP and interleukin 6. Traditional inflammation tests such as serum CRP or interleukin 6 measurements provide a snapshot of your inflammation as a single moment. While helpful, these tests are often influenced by temporary factors like infection or stress, making them less reliable for assessing long-term inflammation. In contrast, DNA methylation-based inflammation tests analyze methylation patterns at specific sites in your DNA. These patterns offer a more stable and precise measure of your body's overall inflammatory state. So, better long-term insights. Unlike serum markers, DNA methylation affects cumulative inflammation over time, okay? It's a bit like your glycated hemoglobin versus your glucose level, okay? It's just a spot in time. More reliable for chronic conditions, DNA methylation, CRP, and interleukin 6 measurements are less affected by temporary fluctuations, making them better predictors of chronic inflammation. Stronger Health Association study shows that DNA methylation, CRP, and interleukin 6 are more closely linked to cognitive function, brain health, and chronic disease risk than traditional tests. Checkpoint number 8, type 2 diabetes risk. While genetics play a role in type 2 diabetes, lifestyle and environmental factors also significantly influence its development. DNA methylation can modify how your body responds to metabolic challenges, such as how it processes sugar or produces insulin. Two specific genes, ABCG1 and phospho1, have been identified as important players in type 2 diabetes risk due to their roles in metabolism and glucose regulation. Changes in DNA methylation at these genes associated with either increased or decreased risk of developing type 2 diabetes. Okay, how this test helps you. Our test analyzes DNA methylation at these two specific locations, providing a personalized assessment of your type 2 diabetes risk. Okay, ABCG1. Okay, if it's higher methylation, it suggests an increased risk in type 2 diabetes and potential metabolic changes. And if conversely, if it's high methylation as phospho1 location, it indicates better metabolic health and a reduced risk of type 2 diabetes. This approach is more dynamic than traditional genetic testing and reflects how your lifestyle and environmental are effectively influencing your genes right now. So it will just give you a methylation score and either a high risk, low risk, or a normal risk. These are the two things they measure at methylation. ATP binding cassette transporter G1, okay, ABCG1. ABCG1 helps maintain cholesterol balance and lipid metabolism. It also supports insulin secretion by removing excess cholesterol from tissues, particularly the pancreas where insulin is produced. Altered DNA methylation in ABCG1 has been linked to reduced gene activity, impairing glucose-stimulated insulin secretion and contributing to inflammation of pancreatic beta isolate cells. That's where insulin is produced, okay, these B cells. This can lead to higher fasting blood sugar levels, increased triglycerides, and insulin resistance, all key markers of type 2 diabetes risk. Research indicates that increased methylation at ABCG1 locus, okay, is associated with a 9% higher risk of type 2 diabetes. This methylation is correlated with metabolic traits like high body mass index, high glycated hemoglobin, okay, HbA1c, and triglyceride levels. So this will give you a methylation score, okay, whether it's high, you have high risk or low risk. And the other thing that's measured is phospho1, phosphatase orphan 1. Phospho1 is primarily involved in skeletal health, playing a key role in bone mineralization. However, it also affects vascular health and has been linked to glucose metabolism and insulin secretion. Changes in DNA methylation at phospho1 influences how efficiently your body regulates blood sugar and produces insulin. Interestingly, higher methylation at this gene has been associated with improved HDL good cholesterol levels and a reduced risk of type 2 diabetes. Research showed that further methylation at higher methylation levels at phospho1 locus is linked to a 15% lower risk of developing type 2 diabetes. And obviously this test will give you a methylation score and risk assessment of type 2 diabetes. So the bigger picture, if your methylation patterns suggest a higher risk of type 2 diabetes, it doesn't mean you're destined to develop the condition. Instead, it's an opportunity to take proactive steps at lowering your risk. Ignoring elevated risk could lead to chronic insulin resistance, cardiovascular disease, and weight gain. Checkpoint 9, okay, it's smoking and disease risk. Smoking is one of the leading causes of preventable disease worldwide. But exactly how does smoking leave its mark on the body? The answer lies in epigenetics. Smoking doesn't harm your lungs or your heart. It leaves a long-term imprint on your DNA by altering DNA methylation, a key mechanism that controls how genes function. The role of DNA methylation in smoking-related disease. Okay, smoking disrupts this delicate balance, particularly a site called AHRR, okay, aryl hydrocarbon receptor repressor. This gene helps the body clear toxin, including those from cigarette smoke. Hypomethylation, that means reduce methylation of AHRR, this gene site, It's associated with smoking, impairing the gene's function. This can lead to increased inflammation, plaque buildup in arteries, and a higher risk of cardiovascular disease. So higher methylation levels associated with non-smokers. So if you have a low methylation at this point, it means you're smoking. Okay. There you go. This is an example of a report. High methylation, so there's no big risk of smoking-related disease. Checkpoint 10. Alcohol consumption is one of the leading lifestyle risk factors contributing to chronic disease worldwide. While moderate drinking is often considered acceptable, excessive or chronic alcohol use significantly increases the risk of conditions such as liver disease, heart disease, cancers, mental health, and accelerated aging. Okay. Your DNA responds to how much you drink. Alcohol consumption alters DNA methylation, a process that regulates gene activity by turning specific genes on or off. Study shows that heavy alcohol use is linked to significant changes in DNA methylation, particularly at genes involved in liver and blood health, okay, and thereby accelerating aging in these tissues. Immune response, lowering our body's ability to fight infections. Cognitive functions, obviously, it would decrease, leading to memory loss and dementia. So, associated gene analyzed in this test for alcohol-related disease. Our report analyzed specific methylation markers at genes influenced by alcohol, including SFSRA13 and etc., etc. Some of these markers have been implicated in liver function and damage from alcohol, cognitive function and brain health, and cardiovascular risk. Analyzing these markers provide a personalized assessment of how your drinking habits have impacted your body on a molecular level. Your fitness age. As we grow older, physical fitness naturally declines, impacting how well our body performs daily tasks and to fight off illnesses. However, the rate at which fitness declines can vary between different individuals. Those who maintain a high level of fitness as they age tend to live longer, healthier lives, and with lower risk of chronic disease. Fitness age affects how all your body feels and functions based on your physical fitness and physiological markers. It's more than a number, it's a measure of how well your body is aging. There you go, you give your fitness age based on your methylation pattern. Your fitness age doesn't just reflect how you feel, it influences your DNA. DNA methylation, which regulates how genes are turned off or off, is directly affected by your fitness levels. And what do they measure? BioFit, okay, your fitness age, okay. For instance here, in this example, 47.25 years old. It's actually 4.25 years older than your actual calendar age, okay, in this particular example. It also measures your gait speed based on your methylation patterns, okay, how fast you can move, your grip strength, okay, epigenetic marker proxy, okay, your grip strength, and then your VO2 max, how much oxygen you can consume, okay, per minute. Checkpoint 12, weight loss response. Why is weight loss different for everyone? Some people find it easy to lose weight, some find it very difficult. The answer lies in your DNA methylation, a process that influences how your genes function. Epigenetics help us understand the molecular factors that may impact your body's response to caloric restriction, offering a personalized view of weight loss potential. This test examines the DNA methylation patterns around the PON3 gene, which plays a critical role in how your body processes fat and responds to caloric restriction. How does the PON3 gene work? It protects lipoproteins, HDL and LDL, from oxidation, which helps reduce inflammation and prevent arterial plaque buildup, and regulates triglyceride production, which is linked to high fat storage. And why does methylation matters? If it's hypomethylation, what you get is a reduced the PON3 gene activity, potentially impairing fat metabolism and increasing inflammation. So if it's a healthy or hypermethylation, it enhances the PON3 gene function and thereby improving your body's response to caloric restriction and fat burning. Okay, this is the last slide. PON3 gene produces a protein that binds to HDL and HDL cholesterol, protecting these molecules from oxidation. It prevents inflammation and build-up of plaques in these arteries, and thereby influences how your body stores fat, and particularly in the forms of triglyceride. Studies have shown that methylation status of CPGI loci, specific DNA sites near the PON3 gene, can predict whether someone will respond to caloric restriction as a weight loss strategy. So, non-responder, intermediate responder, and a full responder, basically based on your methylation patterns. Okay, so methylation, hypermethylation of PON3, related CPI locations suggest that caloric restriction is effective in weight loss for you. Increased methylation may improve fat metabolism and reduce inflammation, optimizing your body's ability to lose weight. So you can see methylation patterns can affect how easy or how difficult it is for you to lose weight. So thank you for listening.